Hello and welcome to the first day of MD and DI's Focus on Fundamentals course, Chemistry for Toxicological Risk Assessment, Transitions and Expected Approach. It's sponsored by Nelson Laboratories. My name is Peter Krass, and I'll be your moderator today. Before we begin, I have a few very quick announcements. First of all, this webinar is designed to be interactive, and the dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media, and participate in the question and answer session that we'll have toward the end of the presentation. The slides will advance automatically throughout the event, and if you'd like to download a copy of the slides, you can do that via the resources widget. Toward the end of the webinar, I'll ask you to complete a short survey, and that's found on the right-hand side of your screen. I hope you'll take a minute to fill out this survey before you leave us today. Your feedback helps us to improve events in the future. And finally, if you have any technical issues during this presentation, please click the Help widget that's at the bottom of your screen or type your issue into the Q&A. And either way, we'll be glad to offer you one-on-one assistance. Let's now move on to our presentation, Chemistry for Toxicological Risk Assessment, Transitions and Expected Approach. Our main speaker today is Dr. Matthew R. Jorgensen. He's a senior chemist and materials scientist at Nelson Laboratories. And we will also have additional commentary and help with some of the questions from Thor Rollins. He's the Director of Toxicology and E&L Consulting, also at Nelson Labs. And with that, I'm going to hand it over to Dr. Matthew Jorgensen. Matt.
Hi, everybody. So thank you for that introduction. And, I couldn't be more happy to be here and talk about chemistry and what's going on in the world of chemistry for medical device toxicology. So it has been a whirlwind of a year for everybody. There's a lot of stuff going on, and in the background of all of the the news and stuff that we're dealing with, there's been a lot of things like changes in perspective with respect to chemistry for toxicology. And so my role at Nelson Labs is kind of unique in the sense that I wear a few hats. I do a lot of toxicological work myself, but I also do a lot of interfacing directly with the FDA for pre-subs that we help out with, and also when the FDA raises questions about chemistry studies that we're providing support on, I also get called in on that as well. So I hear a lot from the FDA. I talk with them, usually more than once a week. So really, what we want to share here is some of the feedback that we've received and kind of give a broader perspective on how the expectations for these types of studies have shifted. Before I get too far into that, let me just, do a little bit more of a personal introduction. I prefer to kind of let you get to know me a little bit better. So my background really is in chemistry and physics, actually more physics than anything else. So I love a good theoretical physics problem and a good math problem. Spent a lot of time working in theoretical physics, doing research before I landed at Nelson Labs and learned all about medical device chemistry and biocompatibility from Thor Rollins. So my main motivation in medical device biocompatibility is to really support the scientific valid alternatives to animal testing, and chemistry and toxicology really provide a window to do that. And maybe my one claim to fame in biocompatibility is just that I know Thor Rollins, right? So he's kind of like a legend in the field. So I'll take that. Outside of medical device stuff, my one claim to fame is that I helped to create a flavor of Mountain Dew that sadly is starting to disappear from the shelves, but because of that and for that reason, a lot of people call me Dr. Dew. And all of you, listening to this webinar are welcome to call me Dr. Dew anytime. So let's get into this a little bit. I think I'm going to start by providing some fundamentals of chemistry for toxicology and even a little bit of toxicology, because that gives us the framework that we need to really have a conversation about how things are changing. So let's just get right into this. If you have a medical device that is going to be submitted for regulatory clearance, then to try to safeguard the health of people using those devices, it needs to be demonstrated that the device is biocompatible before a regulatory body is going to approve it, right? And this is just to prevent these devices from harming somebody rather than helping them. And depending on the nature of the device that you have, there's all of these different biological endpoints that need to be evaluated. So the FDA will say if you have a permanent implant, okay, we'll prove that this doesn't cause genotoxicity or prove that this doesn't cause some sort of chronic toxicity. And the nice thing about this list of biological endpoints, so all of them have a good reason to be on there for something like a permanent implant. Several of those can be considered a systemic toxicological endpoint. So these are adverse effects that could happen related to chemicals that maybe are released from the surface or that migrate through device materials and then are distributed systematically throughout the body, carried by the vascular system or the lymphatic system. So those effects are related or could be related to systemic distribution of chemicals. And of those, there's a subset that the FDA and other regulatory bodies have clarified that directly that they'll accept chemistry and toxicological risk assessment to address those points. And so really what that allows us to do is a couple of things. So first of all, we can avoid the use of animals for things like acute systemic tox. And I really value that, because if you think about something like a titanium, like a TI6AL4V knee implant, we've tested titanium over and over again in Nielsen Labs. We know that there's not an acute systemic tox effect related to that. I really hate to test that again in animals. So it's nice that it gives us an avenue to avoid unnecessary animal testing. We can do this in a way that I think is scientifically a lot better, a lot more sensitive across the board for these. So that's it's a great opportunity to do better science and spare animal life and be more protective of patient health all at the same time. Okay, so how do we actually do this? So what's the role of toxicology in this, in making a claim that something doesn't cause chronic toxicity? And to try to get into that, I'll go into my one slide intro to toxicology. And so really toxicology seeks to answer what is kind of a fundamental question, like how much of a good thing is too much of that thing? And in making that type of determination, we have to consider things like the route of exposure and duration of exposure and potential negative outcomes and so on and so forth. And when I think about this and I try to explain it, it's easiest, I guess, to use myself as the guinea pig example. So as you might imagine, having helped to create a flavor of Mountain Dew, I also like to drink quite a lot of Mountain Dew, and so you can think about like the exposure of myself to caffeine and what that might do to my system, And so I plotted this out, trying to be as fair as I could. Now, certainly since I've built up a tolerance to caffeine, I've reached a sort of homeostasis where if I have no caffeine, then I think that there's going to be some adverse effect there. So definitely going to be irritable. Probably there's going to be some headaches and other negative effects of having none. And in some ways, it's like other things that, not caffeine, but that we need to live, so like essential vitamins and minerals. So there's also like a negative effect to not having any of those. And then you can see there's this well or this minimum where, it's like the least amount of adverse stuff as possible. And that's, my new normal. This is my 3 to 4, Mountain Dews per day. That's like right there. And of course, as you continue up on ramping up the doses, then you start to see all sorts of other adverse effects coming into play. So there is a point where you can have so much caffeine that you start to have like heart arrhythmias, right? So it would be kind of like the next thing up on the list. Then you get tremors, and you keep on going. You reach a point where hallucinations are noted. And then there's kind of like a special point on this curve that's representative of a 50% chance of death. So that would be what we call the LD50 for a compound, the lethal dose, where 50% of the population is expected to have mortality. And then eventually you always can reach a point where you don't see any change in the dose response anymore. And in this case, for example, so at about 150 milligrams per kilogram, caffeine is like 100% likely to be fatal. And so that's about the amount of caffeine in 100 Red Bulls for somebody that's my size. So it should eventually plateau off. If I consume 600 milligrams per kilogram of caffeine, there's no difference between that and if I took 150. So what we want to do is apply these types of curves that where these have been measured in animals over the last 50, 60 years. They've been dosing animals with progressively higher doses of chemicals that are either used or found in like packaging materials for food contact things or as impurities in pharmaceuticals. So there's been a lot of work to generate dose response curves, and all of these are available in the literature. A lot of it's like published in big databases like ECHA and so on. And normally the curve really looks like this. And what we look for in the process of doing toxicology is we look for these two key levels. One is the highest dose where nothing bad happened. That's the no observed adverse effect level. So that's the highest amount that they dosed to an animal model and nothing bad happened. And then at the same time, we also look for the lowest observed adverse effect levels. That's the lowest dose where something bad happened. And I usually try to find studies when I'm doing toxicology where there's both a no-L and a low-L. So the presence of a low-L tells me that the system, the experiment was capable of seeing an effect, right? So, and then the no-L tells me that the highest dose that it would be protective. And in addition to these like more fundamental toxicological data points like no-Ls and low-Ls, there's also a whole like slew of government mandated thresholds and like RFDs and PELs and so on. And these are really based on no-Ls and low-Ls published in the literature, but then government toxicologists have reviewed those and then using uncertainty factors set these thresholds. So we'll often use government mandated thresholds as well. Okay, so that's kind of like a super fast introduction to toxicology. We have all of this data out there. What happens when systems are exposed to these chemicals. Now, if you think about this in the context of medical devices and medical device biocompatibility, if we want to use that information that's in the literature out there, what do we really need to do? So one of the things that we need to do is we need to be able to predict one way or another what chemicals might leave a medical device and then contact the body, right? So we need to be able to do that, and we also need to know, how much of those chemicals are there. So it's not enough to do like a true or false thing and say yes, there's DHP present. We need to know that there's DHP present and how much of that is there. And when I say how much, I'm really thinking of it in a kinetic sense, where we're thinking about how much per day might be released by the device, because all of these this tox literature, it's based off of total amount per kilogram per day. So that's the type of information that we look for, and that's exactly what we do when we conduct chemistry for medical device toxicology. We always have that in mind. It starts with this toxicological goal, and then the chemistry is kind of like a means to an end. So the best way to do that, I think, is with an example. This is one of my favorite examples because it's, this is a real medical device. That's a 3D printed spa can that's intended to be implanted in the body next to the heart, and that way the patient can keep the memory of Leonard Nimoy with them always, right? And so like when you think about this as a device, then then you start to wonder, what would you be concerned about if you were the recipient of it? And it's not something that's on just a targeted list, though there's lots of targeted lists of stuff that might be concerning. It's really everything that's on a targeted list, but it should be a completely open scan and screen type of approach to look for what might not even be expected on there. And this word screening is really kind of like a buzzword. It's throughout the new ISO 10993-18. It's mentioned many times, and I think that's really important to remember as kind of like a seismic shift in the attitude of how are we supposed to do this chemistry from today to 5 years ago, is that we've really changed to a focus on these open screens and doing these open screening type studies in as high a quality of a manner as possible. And so just as a baseline, if that's our goal, to do a huge open screen and look for as many different things as possible, then that really frames the whole problem. And to accomplish that, we need a suite of analytical methods and extraction methods to make sure that we capture as many different things as we can. The analytical methods that we have on hand that are capable of this type of thing are all chromatographic methods. So we use a lot of GC and liquid chromatography, ICP-MS and ICP-OES for inorganic compounds. And using these tools, we create wide open screens, and we look for as many different things as we can. It's important that we really have a complete set of analytical techniques available so that we don't miss entire classes of compounds. So for example, it wouldn't really be good to do this without using any LC, right? Because then even if we did a great job with GC and with metals, we know that there's like really big non-volatile molecules, and we would miss those. So it's important to have a complete set of instrumentation. Certainly the FDA and other and notified bodies in Europe are now very well familiar with the suite of analytical methods that should be used. So I would say that's something that has changed actually over the past 6 to 12 months, is that there's been really a hyper awareness on the part of the FDA about the scope of these methods and what at like a minimum should really be included. So I'm showing this slide again, and then we'll get into some FDA feedback here in just a couple of slides, focusing on how things are different. So just to remember, we're going to do this whole suite of analytical methods, but we have to do it in a way that we can create data that's sensitive enough that we're sure that we can compare it to the toxicological information in the literature. So the sensitivity of this whole process is super important. It doesn't make sense to screen for everything, but then set the sensitivity super high so that tons of stuff are just missed because they're below the sensitivity, right? And so we've begun to hit that sensitivity question head on. It's called in 1093-18, it's the analytical evaluation threshold. So this is the sensitivity that's required for these analyses, so that we know that we're not missing anything that might be really concerning. And this analytical evaluation threshold is based on the TTC concept, which is the threshold of toxicological concern. So we have a new standard, relatively new. It came out in 2019. This is ISO 21726, and this really formally applies the idea of the TTC to medical devices so that we can use it to make an analytical evaluation threshold. And in here, you can see I just reproduced the table from the document that highlights the different thresholds of toxicological concern depending on the duration of contact of the device. So the reason why this is important is before you even start doing the chemistry work, you need to know or have an understanding of the contact type and contact duration for the device so that you can pick the right TTC, so that you can pick the right sensitivity of the study. And the FDA expects all of this to be worked out, prior to initiation of a chemistry study, so that you can be sure that you're hitting the right sensitivity. The other thing that I want to point out here is that a lot of folks and labs, and I guess I would also say toxicologists, they just sort of like expect that the sensitivity is as good as 1.5 micrograms per day. So that's the lowest TTC that we have. You can see it there in the greater than 10 years column of that table. I just want to note that 1.5 micrograms per device for some types of devices can be really, really challenging to screen that low. And really to do the AET right, you need to go even lower. And so it's not always the wisest or most appropriate thing to use the absolute lowest TTC. Rather, it makes more sense to choose a TTC that matches the actual contact type that you have for your device. So for example, if you had a resorbable device, let's say like a resorbable suture anchor, something like that, then even though it's definitely longer contact duration than 30 days, which would put it into the long-term category according to ISO 10993-1, it still would also be known that it resorbs in less than 10 years. So then you can use a higher TTC than 1.5. You could use a TTC of 10 to form the basis of your analytical evaluation threshold. And so this analytical evaluation threshold, like I said, it's core to this whole conversation. It's a little bit new in the sense that within the last 6 months, I mean, this articulation of what is the analytical evaluation threshold and how is it applied, I really see that as an absolute requirement for submission to the FDA at this point. So if you don't articulate that, then you can expect a question and delays related to that. Just so that it's totally clear, so the way that this works is we'll take a look at the contact duration of the device. We'll say, okay, the TTC for this device category is 120 micrograms per day. And then we'll say, okay, we need to make sure that there's nothing from the device that would come off at a higher amount than 125 per day. And so we'll set a threshold based off of that. But we also know that when we do screening, the way that we estimate the concentrations based off of peaks before we know what that compound is, there's a little bit of uncertainty in that estimation process. So we have to account for that uncertainty in our estimation of these concentrations in the screening process as well. So we'll say, okay, the TTC is 120 micrograms per day, but when we do an open screen and we don't know what the concentration, we don't know the identity of those compounds yet, then we think, okay, that concentration estimation is good within a factor of 4. So actually, we'll set that threshold at something like 30 micrograms per day rather than 120, and that way we can be sure that there's not anything that we're skipping that might be present at more than 120 micrograms per day. And this is what we've seen that the FDA wants. So this is the equation that represents what I just described, where the TTC is this dose-based threshold, and you can see here's the copied and pasted feedback from the FDA. word for word. So there we had used this uncertainty factor of 4, and there, not only are they expecting this to be articulated, but now they're asking questions about this uncertainty factor of 4. So I'll just briefly read part of this. So it says, we ask you to justify this uncertainty factor of 4. We replied by saying, or the sponsor replied by saying, yeah, this factor of 4 comes from the literature, these Jordy and Jenke papers. However, the FDA said even though the literature has supported an uncertainty factor of 4, we really think that you should tell us what your uncertainty factor is based off of the data that you have. So it says, although you used a conservative factor of 4, you didn't provide any data to justify this. You should provide calculations you made based off of the relative standard deviation to justify this. So not only are they expecting it, but now they're like getting into the really nitty-gritty about, how are we using this uncertainty factor? What data really supports that? And even though a lot of folks have used an uncertainty factor of 4, based off of information in the literature, I'm seeing them want, more lab-specific data to support this. I mean, I guess the good news is that these uncertainty factors of 4, that's pretty conservative. If you take a look at those papers, I think that's kind of an overestimation. Most often I've seen that when, we've really drilled down on what the true uncertainty factor is for a certain class of compounds or for a certain type of experiment, it's usually a lot less than 4. So a factor of 2 is a little bit more common. I've seen studies where even less than 2 can be scientifically supported. So in the end, it's good because it, makes the threshold that we have to hit a little less burdensome. But at the same time, we have to be prepared to provide or supply that additional detail that so far we haven't had to in the past. Okay, that's this one key shift in expectations. This threshold is super important. Be ready to defend what this analytical evaluation threshold is and how you got to that specific number. The wisest thing to do would be to do that up front, probably in your pre-sub. If you don't go through the pre-sub process, then I would for sure make sure it's included in your chemistry reports and in your toxicological risk assessments to avoid getting kicked back on this point. Okay, let's talk a little bit about choice of extraction vehicles. So this is something that I think about a lot. There's been a lot of conversation, and if you've submitted stuff to the FDA, it's really common to get feedback or questions on the extraction solvents that were used in a chemistry study, and why those solvents, and is it conservative enough, and so on. I usually like to start this conversation by talking about different measures of extraction solvent polarity to try to frame this in a scientific way. So there are a couple of different ways to measure the polarity, the relative polarity of a solvent, including some colorimetric ways and so on. But the way that I really prefer, and this is borrowing from strategies used in pharmaceutical compounding, is to use the dielectric constant of the solvent as a measure of solvent polarity. So you can see here the dielectric constants of lots of different solvents plotted out, and there's really a direct correlation between dielectric constant and solvent polarity. And the nice thing about using this as a measure of solvent and mixture polarity is that not only is it possible to measure for a bunch of different solvents, it's also really possible to measure directly the polarity of different tissue types and mixtures, right? So you can see here, water has a dielectric constant of 80. We know what the dielectric constant of blood is. I mean, it's something we measure. It's ranges between 70 and 75 depending on the individual and their diet and a bunch of other stuff. If you think about like the least polar tissue that we have in our body is probably brain tissue. This has a dielectric constant ranging from 43 to 58, And then you have other solvents commonly used for medical device extractions, IPA at 18.3 and hexane all the way down there at the end with 2. And so I would say, looking at this slide, if we want to be able to bookend the type of polarity environments that a device might contact in the body, really only 2 solvents should be necessary for that. For sure, we have to have water, right? Because that's the most polar thing that we have in our body that these devices could come into contact with. It's more polar than blood. And then I would say we definitely need something less polar than body tissues and blood. And for that, I would say IPA and hexane, from a scientific perspective, make the most sense. Of course, I think we understand that the expectation from a regulatory perspective is that you really should have at least 3 solvents, and it used to be 3, a polar, a midpolar, and a nonpolar, for just permanently contacting devices. I've seen the FDA really want that even for prolonged and in some cases limited contacting devices. And part of the reason for this idea with limited contacting devices is that there's a perspective out there with some FDA reviewers where if a chemical can leave the device, it could remain in the body for longer than 24 hours, and then that would push it into the prolonged or maybe even permanent category, absent of any additional information to support that. My response to that is always, wait a second, when we're talking about contact duration, it's really about exposure, not the pharmacokinetics of the stuff that leaves. But still, I think we all should be aware that the FDA is wanting to be very conservative when it comes to the extraction matrices that are used, and that this is complete as possible. I think that for anything that's prolonged or permanent, you should anticipate at least attempting 3 solvents, a polar, midpolar, and nonpolar. And then if for some reason you want to exclude one of those solvents or one solvent is not compatible, you really should be prepared to provide a rigorous scientific justification on why that you're not using that, and just understand that there's even if the scientific argument is good, really the policy and expectation is that you have 3. And part of this is like built into the new ISO 10993-18. You can see here there's different recommended extraction conditions inside that document. Exhaustive extractions with the 3 solvents are recommended for prolonged and long-term contact devices. Limited contact devices, at least in the standard, are they have like a lighter recommendation with either simulated use or exaggerated conditions. I think that the expectation within the FDA is that these extractions should be conducted exhaustively. Is one of those things that we've pushed back a lot on over the past 2 years. So this exhaustive extraction thing has kind of been going on for some time. It's my perspective that it's really it doesn't make a whole lot of sense to me as a toxicologist to require exhaustive extractions, because I don't care so much about the total amount that might come from a device. Really what I care about is the kinetics of how much may come out per day. And it seems a little bit counterintuitive to measure the total amount, like to go through all of this effort to say that, okay, this is the most that we can get out, when really we're going to like disregard that it's the most that could come out and just go to calling that the total amount per day. But one of the things that we have to be ready for is that this is an expectation that exhaustive extractions are conducted for prolonged and permanent devices. You might even consider it for limited devices, even though I don't like it. For every study that we advise on and that we plan out, if they're doing chemistry for a new submission to the FDA as part of a 510K, we really recommend exhaustive extraction every time, just to avoid the delays associated with trying to explain about this. We've seen that NMR and NVR is the most common way that people demonstrate exhaustivity, even though, again, I think that we agree that NVR doesn't have a sensitivity that's really good enough to do that. So that can still be used, but we've also seen other methods used to demonstrate exhaustivity. For example, TOC, THC, and ICP-MS. This is something that I would say we use maybe 1 out of 5 times to show to prove exhaustiveness rather than NVR. We've also seen folks doing like a full ENL workup. I shouldn't say ENL, full extractables workup at 24-hour intervals, where they really analyze each 24-hour extract by the whole suite of analytical methods, and they keep on doing that iteratively until the amount detected of each of these compounds is less than 10% of either the initial or the maximum. So that's kind of like a more burdensome approach in terms of analytical chemistry, but at least it's scientifically quite a lot higher quality. And if you do that carefully, you can kind of get 2 birds with 1 stone there, because you can demonstrate the exhaustiveness and generate the same quality of data that you need for a toxicological risk assessment at the same time. So even though it's more work, you can save some time that way, as opposed to doing NVR followed by an extractable study. One of the pieces of comments that we commonly see from the FDA on this in both the pre-subs and in other feedback is they say something like what you see on your screen there, that we recommend that you provide a tabular comparison of the total amount of extractables obtained by NVR and the combined total amount of extractables observed by GCMS, LCMS, and ICP-MS. And then, the rest of that explains that, if those 2 numbers don't agree, then they're worried, right? So when you do exhaustive extractions for these studies, if you use NVR, then I think that it makes sense to anticipate this comment and provide this type of comparison up front. It's really unlikely that the amount that you see by NVR matches what you see by GCMS, LCMS, and ICP-MS. In fact, it's almost impossible that those 2 things should really agree with each other. But if you know that this is on the FDA's radar, you can provide that comparison and explain why those things really shouldn't agree up front. So that's something that I would highly recommend. Another thing that they've been asking about or had a new concern about is there's now a lot of interest and focus on the FDA's side on all of the sample preparation part of this chemistry process. So I think that prior to more recent times, they kind of this whole chemistry process from extracting devices to results was kind of like a black box. But now they've been digging into some of those details, and it's really common or essential, really, that when you conduct these studies, you extract the devices in water, but water's not compatible with GCMS. So you extract the devices in water, and then you have to do a liquid-liquid exchange to get all of those organic chemicals out of the water and into an amenable solvent like methylene chloride. So this has been happening forever. There's lots of very rigorously studied and validated processes for performing this liquid-liquid extraction. But now the FDA is really wanting to see that data and have some sort of evidence to support that these processes are good and that they're working, right? So for example, here, they're talking about the liquid-liquid extraction and solvent exchange. They say that they don't like that, although there really isn't a lot of alternative there. And then they say, if you do have to do this, then please provide spike and recovery information to indicate the efficiency of this step of the process. And so this is something that I would say, again, it's new. It's a new requirement in the sense that they're asking for information that they haven't asked for before. But we can get ahead of that and we are getting ahead of that by simply providing some of that method suitability information up front as part of these chemistry reports. So these chemistry reports are getting longer and longer as we add all of this sort of like background information to them. But I think it's better to provide this type of background information to the reports rather than deal with questions and deficiencies down the road. And so really when I say adding this type of information, it's becoming more and more like the type of information that's routinely included in environmental work. So we're not doing environmental work in the medical device chemistry business, but these environmental chemistry labs, they're used to, and they've been doing for decades, providing this sort of method suitability and like evidence that the results are true as part of their reports. And usually this is in the form of things like laboratory control samples that have been taken through the entire analytical process, and then they report what the recovery was for those compounds. I really see the FDA starting. They don't want environmental type results or work, but they want like broad screening work with evidence, method suitability evidence, like an environmental lab. So I would say let's prepare or have our expectations aligned that we need to give this information just so that we don't have any surprises down the road. Okay, so with that, I'm actually near the end of my slides here. I really wanted to save plenty of time for questions in case these are coming up. I would much rather talk about the things that you guys have questions about, so that you're at least getting targeted information. Really, I've kind of like speed talked through one tiny part of this entire process, and this process is in itself just a small part of the entire medical device landscape there. So I want to take a second to just point out that we have lots of more educational resources, not only for Nielsen Labs, but also on a broader sense with Sterigenics for the sterilization processes. The content that I just talked about has, we regularly do this over 3 hours and 6 hours, so there's really a lot there that can be discussed. If you're interested in learning more, you can go to look at the resources on the slide and feel free to follow up on that. Of course, Thor and I, we're nerds at heart, and it just makes me so very, very excited when anybody emails me, because I just finally get to talk to somebody. Certainly, if you have any questions that you don't really want to, ask in this kind of public forum, feel free to email me or Thor. We're always happy and eager to help. And with that, I think we'll transition over to the question and answer part of this time.
Okay, Matt, thanks. This is Peter Krass, your moderator. I'm back. We are going to do Q&A next, but before we get to that, I want to remind everyone about the webinar survey. I mentioned that earlier. It's on the right of your presentation window. If you close the survey, you can reopen the widget. Just click the icon along the bottom of your screen. Once again, thanks in advance for filling out the feedback form. Your participation in this survey will help us to serve you better in the future. And now let's move on. Let's go on to our question and answer portion. It is not too late. You can still type your question in. Just type it into the text box that's located to the right of the presentation window. Or you can click on the Q&A icon that's at the bottom of your screen. And we'll try to get to everyone's questions. If we don't have time to do them all, we'll certainly share your questions with Matt and Thor, and they can reply to you offline. So let us go right into our questions. First one comes from Russell, who asks, can you please discuss simulated use and also determining the maximum daily release sequentially?
Yeah, sure. I'd be happy to talk about simulated use. So this is something that I see as a really important point here. So when we talk about medical device chemistry for toxicology, it's really common for us to say medical device E and L. And even though I'm guilty of saying that a lot, really the L part of that is an abuse of the English language, right? So we can do medical device extractables, but we really can't do leachables because we can't measure these chemicals as they go into actual biological tissues. So what we do instead is simulated use, or we attempt as close as we can in an analytical setting to simulate the conditions of being in the body, and that means that we would do something like 37 degrees C and a solvent that most closely mimics the tissue environment that the device would be in. Commonly, that would be water, or 40% ethanol in water. There's a couple of different mixtures there that really make sense. So simulated use means to try to target actual clinical use with the understanding that that we're never going to be exactly like a leachable, and then determining the maximum daily release. So often what we'll do when we do a simulated use study is we'll use clinically relevant conditions for 24-hour periods, but then we'll sequentially extract the device in fresh solvent over multiple 24-hour periods. And what you expect to see in those conditions is that during the first 24-hour period or the second 24-hour period, it's likely that the maximum daily amount will be there, and then you see the amount per day taper off as the surface of the surface volume of the device becomes more and more depleted of potentially migratable compounds. And so what we'll do is we'll say, okay, to be conservative, we'll take whatever that maximum amount that we observed migrating from the devices and call that the chronic daily lifetime exposure. So that's really the way that we do it, but usually we do that with evidence that it tapers off over time.
Okay, great. We have a lot of questions. Okay, here's one from Janelle who asks, are you aware of any government-mandated threshold for NVR residues?
Yeah, that's a good question. That's a good question, Janelle, and I'll just say from the beginning, I really don't like NVR as a scientific method. I hope that there really aren't government-mandated thresholds because it's just such a poor analytical tool. That said, I'm not aware of any government-mandated thresholds. The closest thing that we have is an ISO 19227. That's the ISO document that sets the standard for cleanliness of orthopedic implants. There, it suggests that an amount of 0.5 milligrams might be acceptable for NVR, but nothing so far officially government recognized.
Great. Okay. Our next question comes from Tuba. Hope I'm pronouncing the name correctly. How do we decide a sample size? Is there a standard for the sample size calculation?
That is a good question, and the short answer is that no, there isn't really a direct standard for that. Oftentimes labs are tempted to use ISO 10993-12, which sets surface area to volume or mass to volume ratios, and that can kind of like fix the sample size in terms of like how much volume is needed for an analysis and so on. But from my perspective, using those types of ratios doesn't make any sense because we don't care about amount per surface area. We really only care about total amount per device. And usually, depending on the known sensitivity of an analytical method, that might mean we should just pool several devices together to make sure we hit that sensitivity. So I'd say what matters most is that we hit the toxicologically relevant sensitivity that's needed. If you mean by sample size like testing in triplicate, we do see people asking about testing in triplicate. Most of the time, that doesn't make a whole lot of scientific sense because the variability in these testing processes is kind of known to be greater than the variability in medical devices.
Okay. Our next question comes from Alexander, who asks about the FDA. Was there a specific event that triggered the FDA's new approach and expectations, or is this just part of an always evolving field of science?
I feel like Thor probably knows the FDA and how this has evolved better than anybody else. Thor, do you maybe want to take that one?
Yeah, so that's a very good question, and actually there's probably both to that. So there's been a few things that's happened over the years. So I've been doing this for 18 years. When we first started, it was pretty much, people would say it's stainless steel or titanium, we don't need to test it. Maybe they do a cytotoxicity test only. But then we started seeing some of these more complicated devices that just didn't fit into the biocompatibility testing. So that had made us kind of think about how we can make sure they're safe. But then we also started seeing things like metal hips that had a big impact. We saw some, the breast implant issues over in Europe and just other devices that started causing problems from a safety standpoint. And we started realizing, and we started evaluating some of the classic biocompatibility tests that we've been running and realizing, like the genotoxicity test, for example, and realizing that they're probably not sensitive for medical devices because we extract them instead of using a pure dose as like a drug. So all of this kind of mixed together, plus the chemistry got better. So the pharmaceutical industry and the food industry was using extractables and leachables to predict safety. And so we started learning from those too. So science progressed, devices progressed, and then also we saw some issues on our historical testing, and so it really was outdated. We really needed to push forward, and so it was kind of a combination of all those things hitting at once that's kind of changed how just not the FDA, but the whole world's now evaluating medical devices for safety.
Great. Okay. Our next question comes from Sharmila, who asks, if a device disintegrates in any of the recommended nonpolar solvents, which solvent is acceptable to represent the nonpolar category?
Yeah, we see this come up from time to time when we have devices, especially things like wound care devices like that. So really with those types of devices, it gets to be pretty tricky, right? So if the device degrades in common nonpolar solvents, let's say, hexane, and you might consider IPA, to be either midpolar or nonpolar, then the FDA certainly would want you to try to use a different nonpolar solvent. But as you go outside of well-known or commonly used nonpolar solvents, then you kind of open up another can of worms where, if the FDA wants data or evidence to support the suitability of the method or the quality of the data, you might be stuck also doing experiments to generate that method suitability stuff. So usually what we'll attempt to do with these types of devices is explain why the nonpolar solvent isn't relevant. We can show some data or information that supports that the device just doesn't disintegrate like what we're seeing when it's used clinically. And, I wouldn't say it works all the time, but at least 50% of the time when we provide that explanation, the FDA sees, is eye to eye with us, and we're okay to use just a polar and a mid-polar. There's still a risk there, though, that they may dig in their heels a little bit and demand a non-polar, and then you have to ask yourself, are you going to go through the burden of generating method suitability data and it might still not work, or should you just then unfortunately reflex to some biological testing?
Thanks, Matt. Okay, our next question comes from Sam, who asks, when unknown compounds are extracted, how do you handle that?
Yeah, so that's a great question. It's something that's there's a lot of talk and debate about what really is the best thing to do with unknowns. Certainly, the smartest thing or the best thing to do is to avoid that situation as much as possible. I think that we have to realize and accept that even though and I am biased as a chemist, even though chemistry is so awesome, there is some limitations, and we're going to have to deal with some uncertainty in these results no matter what. So even if we see some unknowns, we shouldn't throw the baby out with the bathwater, so to speak. Usually what we do with unknowns is first we can go back to the lab and ask them, can they take a little bit of a harder look at this and consider the materials of construction and so on, and use like supplemental sources of information to try to make at least a tentative ID on that compound. So that's one strategy. Another strategy that's commonly used is to try to look at the exposure and ask if that is truly above a TTC. So it could be that a very conservative TTC was used in the chemistry, but when you actually look at the exposure a little bit harder, that a higher TTC could be used, and that compound would be below the higher TTC. So that's something that we'll do. We'll take a look at some kinetic stuff and maybe using the Arrhenius equation. If we take the data and the extraction conditions that we have, we can predict what the daily dose would be in the body, and if that drops below the TTC, then it's okay. Finally, if there's an unknown that's above, that's too high, and we can't figure out what it is, and it was an extractable study, we might look at doing a simulated use study and see if, under more realistic conditions, if it drops below a threshold of concern.
Thanks, Matt. The next question comes from Tanya, who asks, first states, you have a statement in your presentation, ignore substances that are impossible or improbable. I think that was on the dose-response curve. Anyway, Tanya's question is, can you explain what you mean by that?
Well, I'm glad that you picked up on that. It's good to know that, somebody's reading these slides. So really that statement comes from several conversations that I've had with regulators, and I don't want to sound like negative towards any regulator. Especially the FDA, they have a tough job and they do really an amazing job. So I'm kind of like nitpicking here, but sometimes they'll ask questions like, how do you know that there's not cocaine on the device, right? Like a material-mediated pyrogenicity like cocaine, and I'll say, well, yes, we didn't specifically look for cocaine, but we know that there's not cocaine on the device because that never comes into play throughout the manufacturing process. There's just no valid scientific reason for that chemical to be there. And so there's actually, lots of compounds in that category that are super improbable to be in or out of medical devices that we don't look for, that would be really burdensome. And another good category is things like specific proteins, like really any biological material from a non-biological device. So we don't do fancy methods to characterize potential proteins from devices because we just don't. We just know that they're not likely to be there. So we're ignoring classes of compounds that don't make sense as a matter of principle, because if we had to truly look for every compound, like biologics and proteins and controlled substances and all of those things, it would be impossible to get the study done.
Okay, looks like we have time for just one more quick question, and I have a quick one from Johan, who asks, what is NVR?
Yeah, NVR stands for non-volatile residual. So non-volatile residual, that's NVR. And the way that study is conducted is that a medical device will be soaked in an extraction solvent like isopropyl alcohol for a certain amount of time. We could say 24 hours at 50 degrees C or 50 degrees C for 3 days. And then that liquid that the device was sitting in, a portion of that will be put into a container and then dried. And everything that happens after the extraction is kind of not super standardized across the industry. So how hot do you dry it and how long do you dry it is kind of not super standardized. And then that container was weighed before and after, and whatever that weight is the NVR.
Okay, thanks, Matt. Well, unfortunately, we're out of time. This has been a great presentation. We have a lot of questions that we didn't get to. We will share them with Matt and Thor, and I hope they'll be able to respond to you offline. Also, within the next 24 hours, you'll receive a personalized follow-up email. This will have details and a link to today's presentation on demand. Please invite your colleagues and peers who couldn't join us today to listen to this event on demand. This webinar is copyright 2020 by Informa Markets. The presentation materials are owned by or copyrighted by Nelson Laboratories. The individual speakers are solely responsible for their content and opinions. Again, I'm Peter Krass. I want to thank you for attending today's course, and please be sure to join us tomorrow and Thursday. Thank you.